Hello everyone, and welcome to this presentation about estimating the causal network of developmental neurotoxicants using PyMC3. I'm Nicoleta, a PhD student in computational toxicology at the Chemoinformatics Research Group of Professor Mark Cronin in Liverpool. I'm also a Marie Curie Fellow of the INFREE-I10 project, which stands for the integration of in vitro and in silico tools for better assessment of chemicals exposure. In this presentation, I'll be talking about a methodology we've been developing to assess individual compounds for their potency of inducing developmental neurotoxicity for a specific biological path. Also, how a Bayesian hierarchical multilevel model can be formulated in PyMC3, and how data issues such as variability, missingness, correlations, and causation can be addressed. Well, before going into the modeling itself, I have to talk a little bit about toxicology. We are exposed daily, consciously and unconsciously, to all sorts of types of chemicals, from what we eat, the type of cosmetic products we use, or any other type of products we daily use, besides the quality of the air that we breathe, or the medicines that we take and that can also lead to adverse effects. Well, the current status in toxicology is that scientists agreed upon the need of new ways on how to actually assess the chemicals exposure. The toxicity testing in the 21st century, a vision and a strategy, summarizes this idea. Besides that, nowadays, we've already started to having all these new novel methods, such as in vitro models derived from stem cells, high throughput techniques, or computational approaches such as probabilistic programming languages that can allow to integrate better all sorts of information in order to actually assess in a better way as well the toxicity. Also importantly is that this type of movement is not only supported and promoted by the academia or industry, but also by other parties. For example, the European Commission, who through the European Green Deal aims to achieve toxic-free environment by 2050. Well, in toxicology, we have a concept that is called the adverse outcome pathway. It is a tool that allows to summarize in a transparent way and also to structure the mechanistic knowledge, the biological type of information regarding a toxic effect that can be induced by any stressors, such as chemicals or radiation. Well, what does it mean? It represents a linear representation of our biological understanding, and hence a chemical will bind to a protein at the molecular level and will induce effects at cellular, tissue, organ, organism, and population levels. Well, this linear LP is considered a unit of development. So we'll use this linearity to help us to structure and to better organize our knowledge. However, if we want to develop predictive type of models, we will rather use a network of linear LPs. Developmental neurotoxicity represents a series of adverse effects that come as a result of exposure to chemicals on the nervous system. These adverse effects represent such as motor, sensory, and cognitive effects. You can think of autism and ADHD, for example. Also, this type of neurotoxicity is considered a silent pandemic. The reasons are many, and one of them is that the current chemical regulations require such assessment only if and when a D&T concern exists. On the other hand, the existing standardized methods are based on in vivo animal tests, and such models are time-consuming, imply high costs, and also can lead to extrapolation issues. Most importantly is that this type of developing brain is much more vulnerable to exposure to chemicals comparing to an adult brain. A report published by the World Health Organization almost 12 years ago mentioned that 5% of the old children under 15 years old have some type of moderate to severe neurophysical or cognitive developmental disability. Well, what's the actual problem, or what are the main challenges? It includes limited and heterogeneous type of data. There is still a lack of mechanistic knowledge. The data that is generated mostly is coming from non-standardized methods. So this type of data is not universally accepted. So there is a really need for development of alternative methods to animals testing for appropriate testing strategies and with a final aim of an appropriate assessment of chemicals exposure. Our approach in tackling as many as possible of these challenges was firstly to understand what is the state of the art in terms of developmental neurotoxicity. And for this, we utilized the existing linear AOPs published in the database, the Wiki of Adverse Outcome Pathways of the OECD. And then that type of network allowed us to analyze it using topology type of parameters and mark which are the key events or nodes mostly connected, and also that can be considered essential key events across the network. This work, together with an expert panel, helped to come up with a simplified adverse outcome pathway network. And this simplified AOP network was the one that stayed at the base for the development of the Bayesian model. And the simplified AOP network consists of that a chemical will reduce brain-derived neurotrophic factor, which is a protein. And then once this protein is reduced, it will lead to decrease of synaptogenesis and decrease of neural network formation. These two key events being essential for a healthy developing brain. Upon the decrease of these two biological processes, it can lead to developmental neurotoxicity, such as impairment of learning and memory and cognitive dysfunction. So this simplified OP network helped us to define the likelihood function of a Bayesian model, but also to actually collect appropriate data. Now, what kind of information we collected? You might have heard about bisphenol A as being quite a controversial chemical. It is widely used in the production of plastics. You can think of plastic bottles, and it is considered an endocrine disruptor. Well, there are lots of research ongoing regarding this category of chemical class endocrine disruptors. Our dataset contained 97 compounds, and bisphenol A was one of it. Well, looking at the chemical, we collected in silico predictions, in silico type of information that came from in silico models. We were interested to understand if a chemical is lipophilic, in what type of tissue you can find it, it is fatty or not. Then if a compound crosses or not blood-brain barrier in order to produce, in our case, neurotoxicity. And then the role of PGP, P-glycoprotein, which is a transporter essential in taking the chemical into the cells. Besides these in silico details, we looked to find out what kind of information is available in regards to BDNF. And for this, we've conducted a systematic review where we looked if there is any epidemiological, in vitro, in vivo studies that analyzed each independent chemical. And hence, the final classification was a binary classification of yes, no. For the other two key events, synaptogenesis and neural network formation, there are already developed in vitro models derived from human stem cell lines or from rodents cell lines. These are two independent in vitro studies. For synaptogenesis, we also applied in a binary classification. EC stands for effective concentration, and it shows the activity of the compound of the chemical in the in vitro system. And besides the activity, an in vitro test will also look at the viability. And for neural network formation, viability has been measured by LDH, while for synaptogenesis, the viability has been measured by ATP. And finally, for the developmental neurotoxicity endpoint, the classification was also a binary yes-no. And it came from several review papers, peer-reviewed papers. They summarized in vivo studies that showed if a chemical is positive or negative. Well, what was our way to go with this? Firstly, we wanted to keep the causal graph that was informed by the simplified version of the AOP network as a series of linear regressions. We wanted to model this causal graph in a hierarchical way because we wanted to predict and to model the predictions of each individual chemical. And finally, we aimed to model this in PyMC free. Before going into the modeling, I think it's really important also to have an overview of what kind of data we actually have in our data set. This kind of overview also helps to understand what kind of distributions, how better to define the likelihood functions. And hence, with this slide, I'm showing you the type of variables our data set contained. So we had categories type of data and continuous type of data. And we saw that there is unbalanced number of chemicals across each category. We also looked at the correlations given by the Pearson coefficient, and it helped us actually to understand how better to group information in order to evaluate our final adverse effect. And if we see, for example, for lipophilicity and details about brain-blood barrier penetration of a chemical, we see that these two can be grouped together. We saw that PGP, P-glycoprotein, activity, inhibition, and if a chemical is a substrate or not, can also be grouped together. We saw that synaptogenesis as well, with the activity and viability can be grouped together, but there is also correlation between activity of neural network formation and activity of synaptogenesis. And here, this cluster represents the neural network formation details about this key event. Our dataset also contained missing values, and we wanted to see if our missingness is informative. And for this, we also looked at the correlations between the missing values, and it confirmed to us that it's important to keep all chemicals, even if a chemical can have missing values. And here you can see that we had missing values at all levels, even predictions that came from in silico models. That's because our dataset contained as well metals and not only organic compounds. And hence, that's why here it's a high correlated result for P-glycoprotein. And then 2 in vitro studies that we have included also had missing values because not the same list of chemicals has been tested in both in vitro methods. And hence, that's why here we see a high correlation for synaptogenesis and here a high correlation for neural network formation. Well, what are the key messages from this analysis? Our dataset contained binary and continuous type of variables. It also had unbalanced categorical data, missing values, correlated data, and we wanted to keep the causal linkages that are informed by the simplified version of the OP network. Now the Bayesian modeling. I'll show you first with a simplified structure of a model. It's always useful to actually check the structure while you wrote your code in PyMC3, especially because it helps to find out if you misspecified in any place. The entire structure in PyMC3 can be checked using this function. This is a simplified version, so we try to draw it and to make it a little bit clearer for the purpose of this presentation. As I said, our data set had missing values, and that's why here you see that we computed this missingness for the predictors from a prior distribution. We also had missing values at the level of our observed variables, so our outcomes, and we computed these missing values from the posterior distribution. And we were interested in a hierarchical type of modeling, and hence we defined a common mean and standard deviation that has been attributed the mean and the standard deviation to a beta for each of the key events, BDNF, synaptogenesis, and neural network formation. Our dataset contained 97 chemicals, and we wanted to understand if a chemical induces or not developmental neurotoxicity and what's the probability of that. I've decided as well to include this technical specification of a model. On the left-hand side, you can see the mathematical specification, while here on the right-hand side, how the model is specified in PyMC3. Our interest was, as I said, to actually compute developmental neurotoxicity. It is generated from a Bernoulli type of distribution, and that's why you see here the tilde sign. And this Bernoulli distribution came from a linear regression, causal linear regressions of each independent key event that has been computed as a theta. Theta is calculated from the data itself, so multiplication of our predictors with beta. And basically this is a deterministic function, and that's why here you see the sign of equal. The missingness, as I mentioned, was also computed, but from the distribution either of posterior or prior. And here you see the details of a prior, how we define it, and how we observed our missing values that has been masked previously in advance before running the model. And then we have our priors and hyperpriors that will allow us to shrink our data and such make the model less flexible, but also much more informative. In PyMC 3, you'll start with pm.model function, and then you'll give a name to your model. I would like also to mention that a trick that it's quite important. Always think of the shape function that is given by the number of variables that you want to include in your model. So here you see the number 6 because we evaluated our chemicals for BDNF, including 6 predictors. Here 2 because for synaptogenesis we only included these 2, and for neural network formation 3 because here we had the minimum value of the effective concentration and maximum value of the concentration besides LTH value. Now, because we are talking about a probability, it's also important to transform in a logit scale between 0 and 1. And finally, once the model is specified, PyMC3 can also make choice for you in terms of what kind of technique to apply to run the inferences, but otherwise you can also specify by yourself. In our case, our data has been run using a NU-turn sampler for the continuous variables and binary Gibbs metropolis for the binary values. Now, after you specified your model, it's also important to check it to be sure that there are no divergences. And one way of doing this is to look at the trace plots. And here I've chosen to plot the trace plots of the hyperpriors, mean and standard deviation for 2 chains. On the left-hand side, you can see how traces has been conducted. And here we can see the actual distances between the way the samples have been run. With this slide, I'm showing you the actual trace plots of the hyperpriors and priors. And the inference step has been conducted for 4 chains, and that's why here you can see 4 lines. And here you find different colors because we had different predictors. So we had these results computed together. And on this left-hand sided graph, you also can see once again the mean, standard deviation, and high density interval, which is the Bayesian credible interval for each of these parameter and hyperparameter. Another way of checking your model is to look at the energy transition plot and at the marginal energy plots. So we have here the Bayesian fraction of missing information. It indicates whether the sample is able to efficiently explore the posterior distribution. It should be around 1. There is no threshold for this. And also the differences between the chains should be as small as possible. Also, you might find useful looking at the Gelman-Rubin R-hat statistics. And ideally, it shouldn't be above 1.01. because that should raise a concern to us. And you can get as well all of the statistics summary, and you can check each independent parameter for its mean, standard deviation, credible intervals. Markov chain standard error should not exceed 10% of the posterior standard deviation. Then we have effective sample size, should be larger than at least 10%, 25% of the actual number of samples. And then the R-hat for each independent parameter. Now, in terms of results, I'll start by showing you the distribution of the compounds for theta predictions. As you saw previously, our data set contained missing values, and that's why we have these 2 categories now, yes, and then we also wanted to see the distribution of the missing values. Not for the developmental neurotoxicity, because for that key event, we didn't have any missing values. And for the purpose of this talk, I decided to rather take a small case study and show the results on 4 compounds that I found interesting. You might recall the bisphenol A that I've presented at the beginning of the presentation. I've chosen also dexamethasone, which is another medicine that you might have heard, especially with the COVID pandemic situation these days. Glyphosate, which is a herbicide, and fipronil, which is an insecticide. Now, bisphenol A didn't contain any missing values. It is known to be a positive developmental neurotoxicant. Dexamethasone has also evidence from animal tests that is a positive developmental neurotoxicant. with negative results for synaptogenesis and neural network formation. Glyphosate is considered a negative developmental neurotoxicant, and there are no data for measuring BDNF levels. And then fipronil is also considered a negative DNT, and it wasn't measured for synaptogenesis. Both, if we go across our simplified DOP network, we can see that bisphenol A has a probability of 1. For dexamethasone, the probability becomes almost 60%. Glyphosate has a probability of 3.4, and fipronil a probability of 50%. Now, one of the questions that I asked myself was, if we look at the actual results, the actual predictions, how these predictions can be used, and especially in terms of assessment of chemicals exposure, or how the risk can be communicated or should be communicated to people. And I'll be interested as well to hear your thoughts, and hopefully we can discuss later and brainstorming on this question. One way of looking at the results was to actually understand if we can select a threshold, and then we can say if a compound has a high, medium, or low probability. And here I'm showing you the results for the same 4 compounds. And you can see that bisphenol A has a high probability, dexamethasone a medium level of probability, glyphosate low probability, and fipronil medium. One of the conclusions that can be formulated out of these predictions is that dexamethasone, for example, has a medium probability level. And if our in vivo test showed that it's still considered as a positive DNT, maybe dexamethasone induces developmental neurotoxicity for a different path. So it needs to be further investigated. On the other hand, fipronil contained much more missing values, and hence it needs to be tested appropriate in order to understand if it induces or not DNT. Bisphenol A can be used as a positive control for other in vitro experiments, and glyphosate as a negative control for the same purposes of wet or in silico type of experiments. Well, additionally to this type of model that we've presented until now, we actually compared our model with 3 different other models that we've defined them as well in PyMC3, and we wanted to be sure that the model that we propose is the best one. Hence, here you see a comparison between our model that contains regressions and multivariate model. The main difference actually between these 2 models is the way the prior is defined, besides the actual purpose of the multivariate way of analyzing data. In our case, the prior comes from a distribution, where here in a multivariate model, the prior comes from a Koleski type of variance matrix. And as you can see, the results are almost similar, and hence we could confirm that our model, for example, captured the correlated data, because Koleski helps a lot, especially if you have correlated data, and both this assured us that our model can capture correlated variables as well. The other comparison that we've conducted was to look at the causality versus non-causality. Our model captured the causal relationships, while the results for the below, it comes from a model that didn't capture causality. And here you can see, for example, for fipronil, who had missing values, that it doesn't contain this large Bayesian credible interval because it doesn't take into account the key event relationships here, the key events and linkages between key events. And the results are quite steeper, so they are not taking into account this uncertainty, and basically it actually depends on the purpose. As we wanted really to capture causality, we managed to do it. And finally, another important aspect was to actually compute the missingness. And we decided to impute the missing values from the distribution. In machine learning, there are all those sorts of classical techniques, imputation, for example, the mean. And here you see this comparison between imputation done by mean and imputation done by distribution as followed in our model. And once again, if you look at the fipronil contain the highest level of missingness, it can give not the best result, and then you might have wrong conclusions about it. So with the imputation by mean, it gives a result of almost 81%. Well, to sum up this work, a Bayesian hierarchical model was developed. The methodology allows to quantify a simplified version of an adverse outcome pathway network that has been formulated based on topology analysis and also by an expert panel. The model allows to assess individual chemicals for their potential effect of developmental neurotoxicity. PyMC3 handles perfectly unbalanced, missing correlated data. And a personal judgment is that the output becomes much more informative and reliable compared to a frequentist setting where the conclusions are based on the chosen p-value. I've also decided to list a few key lessons learned during the modeling itself. I would highly encourage you to always check and write the correct shape function. Also use the check test point function that allows to understand if you misspecified your model. Think if you really want to delete the missing values, maybe missingness is informative and you want to keep it. And hence, in a Bayesian setting, you can compute the missing X predictors from the prior distributions while missing come from a posterior distribution. And regarding the choice of priors, that was one of my concern initially, is that just start with your choice. Of course, if you have more data and you can make your prior more informative, it's better. However, you need to understand that the posterior actually allows to compare your judgment, the way you define your prior before and given your data. Here it is a list of essential resources that were useful to me. I highly encourage you to check and read the latest book of Gelman et al. There are a few chapters on causality as well. And definitely check the PyMC3 community. There are lots of resources on GitHub, on their official website, on Discourse. I would like to acknowledge all of these scientists I had the privilege to work with. I would like also to thank the PyMC3 organizers of the conference who accepted and allowed me to come and present this work. My special thanks go to Dr. Yupeng Lango, who is a developer of the PyMC3, who encouraged me and guided me through the process. I'm very thankful for his time and all the suggestions. I would like as well to thank my supervisor, Professor Mark Cronin, and our research team from Liverpool. And I would like as well to thank Dr. Andrew Orff and Annabelle Price that motivated me to go ahead with the modeling and actually to focus on developmental neurotoxicity. I would like to thank as well my colleague Liadis Mora-Lagaris from the IMPRI project and Professor Mariana Novic from the Chemistry Institute from Ljubljana. Their contribution was that we saw the value of understanding the role of P-glycoprotein transporters. And I would like as well to mention my participation at an workshop organized at the Lawrence Center in Leiden, where I had the privilege to be part of a team with Dr. David Pamias, Dr. Dan Villeneuve, Dr. Ivana Campia, and Dr. Stefan Schultz, where we have discussed this conceptual framework that helped do the patient modeling. And finally, I would like to thank you, all of you who have chosen to watch this presentation. I'll be happy to take any questions and I'll be happy to talk to you on Discourse or any other way. I wish you a pleasant day and stay safe.